So, assuming myself audible, myself is Parveen, and I welcome you again on session 2 of this Ensemble Genome Browser Workshop, jointly hosted by Ensemble Outreach Team and decodelive.org. So, we have again with us energetic Michelle Speck, who will lead today's session. Over to you, sir.
Thank you very much for opening the session. Let me just share my screen. So I hope everyone can see my slides now. Welcome everyone to the second day of our Ensembl workshop. My name is Michał Szpak, and today we're going to talk about genes and transcripts in Ensembl. Just a short recap. Last week, we talked a little bit about general introduction to Ensembl. We talked about genome assemblies and the species data you can find in Ensembl. And today we're going to be moving to our gene annotation pipelines, and we'll be talking about how you can find and access genes and transcript data in the Ensembl genome browser. And just a small reminder, maybe for those of you who are new this week, this module will consist of a first small lecture, so a small presentation where I'm going to introduce our genes and transcription data and our pipeline to produce our annotation. We're then going to have a demonstration when I'm going to show you how to navigate the Ensembl genome browser and how to get the data you're interested in. And at the end of today's, we're going to have some time for your own exploration. So we're going to have some exercises. It's basically time for you to go and explore on your own. You're more than welcome to go beyond the demo, use your own data and examples. And yet again, please remember, it's not a test. We provide a nice selection of exercises. Don't feel like you have to do all of them. Please feel free to choose the ones that are more relevant to your work and your interests. And just as last week's, you can find the course materials at training.ensembl.org/event. You'll find there a copy of this presentation, your course book with the demonstration walkthroughs and your exercises and answers, but also the living document, which we use to capture questions throughout this session. So let me just jump out of my presentation to show you those resources. So if I go to my internet browser and if I type training.ensembl.org, and that's going to take you to our training page. And then in the right-hand side, sorry, left-hand side, yes, that's left. And the left-hand side, you can find materials from training. So if you click on that link, you then have to find the event you're attending. So at the top of the page, we've got Ensembl Browser Workshop Decode Live. So if I click here, and you'll find the copy of this presentation, your course book with demos, exercises. Don't worry about the copy-paste file. You're not going to need it today. And then we have this living document. So yet again, you're more than welcome to use the Zoom chat box. However, it gets messy quite easily. It's difficult to keep track of. There's no threading. It's not easy to paste any screenshots. So I would really encourage you to start using the living document. It's pretty much just a shared Google Doc, which everyone can access. So please watch out and mind not to accidentally delete other people's content. So we've got a couple of welcome instructions, also useful links to training materials. And we have some golden rules for online training. So I would ask you to keep your microphones muted throughout the lecture, just to keep the background noise to the minimum. But then if you'd like to ask a question out loud, you're more than welcome to unmute yourself. Feel free to jump in if something's not clear, if I'm speaking too fast, if something doesn't make any sense. You're more than welcome to unmute yourself, ask a question so that we can sort it out as we go. And finally, here we have this dedicated space for questions. So you can see we've got plenty of really nice questions, and I'll be checking out this document throughout today's session. Okay, great stuff. Are there any questions? Let me just maybe copy this link and paste it in the Zoom chat box for everyone. All right, without further ado, let me just switch off my camera now. And go back to my presentation. So as I mentioned, today we'll be talking about genes and transcripts. So here I'm showing you the region in detail view, which we explored last week during our demonstration and exercises. So a gene is a collection of transcripts in Ensembl. And I'm showing you a region in detail view. I'm showing you the ABCC11 gene in particular, and all of the transcripts of this gene are highlighted in green. So you can see all of them all together, they make up a gene. So what information can we learn from that image? Well, first of all, you can see that this gene has multiple alternative transcripts, and that's absolutely normal for multiple transcripts to be transcribed from a single locus. Alternative transcripts are also known as isoforms and splice variants, although personally I have to say I'm not a fan of the splice variant term because that indicates that only alternative splicing is the sole reason behind alternative transcripts, but we know it's more complicated than that. There are alternative transcription start and end sites, there are alternative translation initiation starts, and so on and so forth. So it's way more complicated than that. And they're all biologically valid and real. They could be expressed differently in different tissues and different developmental stages. So what can we learn from this image? Well, first of all, the transcript biotypes are color-coded. So we can distinguish between protein coding genes, so genes which are translated, so they result in a protein, but also non-coding genes, so non-protein coding genes, which are transcribed, but are not translated, like, for example, RNA genes. And transcripts of non-coding genes are shown in this lilac color. So when it comes to protein coding genes, their protein coding transcripts, which are translated, are shown in red. But protein coding genes can also have non-coding transcripts. So transcripts which again are transcribed, but are not translated, like, for example, some retained intron transcripts or nonsense-mediated decay transcripts. So this could be fragmentary, so they might be missing bits, they might be missing translation initiation sites. They could be faulty, so they're degraded before they get translated. And those transcripts are shown in blue. And we also have some called merged transcripts, which are shown in golden color. And these are basically transcripts of higher quality and higher confidence in their annotation, but we're going to be talking about those a little bit more in a minute, so don't worry too much about this for now. So what else can we learn from that image? Exons are shown as boxes, and then non-coding exons are empty. And of course, transcripts of non-coding genes or non-coding transcripts of protein coding genes are going to be composed of empty boxes as they're not translated. And then we have filled or colored exons. So you can see these colored boxes indicate coding exons. So those are translated and they code for protein. However, protein coding transcripts can also have empty boxes at either 5' or 3' end. So what are those? Those are pretty much UTRs, so untranslated regions. Please bear in mind that UTRs are exonic as they remain in transcript after splicing, but they're not non-coding as they're not translated. So not all exons are coding, okay? And introns are shown as lines connecting the boxes. And there's a couple of hints here indicating the gene orientation. So first of all, the gene orientation, or the gene strand, if you will, is indicated by the relative position of a gene to the genome assembly. And forward-stranded genes are shown above the context, so above the genome assembly, and the reverse-stranded genes are shown below. Furthermore, this little arrow next to the gene name also indicates the gene orientation and the direction of transcription and the 5'3" direction pretty much. So for forward-stranded genes, you can see that this arrow is pointing towards the right, while for the reverse-stranded genes, this arrow is pointing towards the left, indicating this direction of transcription. And the final evidence is the shape of introns. So as you can see, forward-stranded introns, they're somehow pointing upwards, so they have this so-called hill shape, while the introns of reverse-stranded genes are pointing downwards, so they have this so-called valley shape. So I hope that's clear. If there are any questions regarding this, please do let me know. And by the way, ABCC11 gene is responsible for the earwax type, as it comes in two types, wet and sticky, but also dry. And just a random fact. So let's talk about the gene annotation in Ensembl. So we use two different annotation methods, two independent annotation methods. First, so-called Ensembl automated annotation, which is applied to almost all vertebrates in Ensembl. And the second one is more laborious, it's called the manual Havana annotation done by a group of curators. And it's only done for human and mouse, and partially for a rat and zebrafish. And they're both based on real biological data. So the Ensembl automatic annotation is a genome-wide gene prediction using our automated pipeline. It's based on real biological experimental data. So what we do, we take known proteins and transcripts, so cDNAs and RNA-seq, and we plot them onto the genome using sequence matching. Okay. So people sometimes assume, they think that our annotation, that we pretty much just screen the genomes looking for open reading frames, but not really. We need real evidence that something is translated or transcribed to make a gene annotation. And it takes about a week or two to annotate a genome using our automatic pipeline. And we take our biological evidence from various sources. So we take most of our nucleotide data from the International Nucleotide Sequence Database Collaboration, which is a collaboration between 3 institutions, and that includes the NCBI's Gene Bank, the DNA Data Bank of Japan, and the European Nucleotide Archive. And from there, we take most of our cDNAs and RNA-seq data. We take most of our proteomic data from UniProt, which includes manually curated proteins in SwissProt and unreviewed translations in Tremble. And finally, in some rare cases, we might also annotate less studied and less sampled organisms based on homology to better studied and sampled organisms. So if there is a very little experimental data and RNA-seq data from a given organism, we might pursue with this homology-based inference. So for example, we could predict genes in chimpanzee by mapping transcripts and proteins from human to the chimp genome. But that's only done for very closely related species. And we, as I mentioned, we then also have manual annotation, which is also a genome-wide gene determination done on a case-by-case basis by a person, so by an annotator who moves along the genome, utilizing all sorts of experimental data, including publications, and it's more laborious. It takes about half a day to annotate one gene, and it might take up to several years to annotate one genome. That's why it's only done for a human and mouse, and partially for a rat and zebrafish for those specific genomic regions of interest. And the annotator can utilize a wider selection of experimental data, including some RNA-seq data, both short and long reads, but also some data supporting the transcript structure, including ACADE and poly-A-seq data, which can tell us something about the transcription start and end sites, but also mass spec, proteomic data and publications. So what are the advantages of manual annotation? Well, first of all, it's more comprehensive, especially for those difficult to annotate non-coding features. So it results in more genes in general, but also more transcripts per gene and more a biotype, especially the difficult non-coding biotypes like pseudogenes, stop codon read-throughs or nonsense mediated decays. It requires less evidence. By what I mean, it could be more sensitive. So for example, if there is a small evidence, the automatic pipeline might skip the evidence without making an annotation, but an annotator can investigate further, can explore further, and can make an informed decision on a case-by-case basis. And finally, it's also more accurate for difficult regions, including UTRs, splice sites, some assembly exceptions, stop codon read-throughs, or single exon genes. The disadvantages is that it's slower, it's smaller scale, so it's less throughput. So now we know that for human and mouse, we use those two independent methods, Ensembl automated annotation and Havana manual annotation. So we have those two independent annotation lists, so lists of transcript models, and for human and mouse, we merge them. And some of those transcript models on those lists might agree, so they could be annotated identically, so they could be agreed upon by both the automated annotation and manual annotation. And if that's the case, we then give them this golden color, which indicates higher confidence and quality of those transcripts. And for a few rare cases, we also import our annotation from external sources, but we don't import it blindly. We also apply our rigorous QC and we make sure we import them from trusted sources. So what is GenCode? You might have come across the GenCode term, which is basically Ensembl human and mouse genes. So it's the GenCode is the Ensembl gene set for human and mouse. It's called the GenCode for historical reasons, but it's pretty much a merged set of Ensembl automatically annotated genes and Havana manually annotated genes for human and mouse. And it's the default gene set used by a major international projects and consortia, such as those listed on the slide. Another important concept is the main project, which is sort of another reassurance of transcript quality and relevance. So a main stands for a matched annotation from the NCBI and EMBL EBI. So what is it all about? Well, I have to start by saying that NCBI has their own gene annotation, their own gene set called RefSeq. And main transcripts are transcripts which are 100% identical between those two resources, so NCBI's RefSeq and Ensembl. And by identity, I mean it's a complete match. So they're exactly the same from the transcription start side until the transcription end side. So a complete match, 100%. And then the main select transcript set provides only one well-supported transcript for every human protein coding gene agreed to be the most biologically relevant of that gene. Okay, so main select transcripts are not only identical between RefSeq and Ensembl, but they're also the most relevant and representative transcripts of a given locus. And by a representative, I mean they were chosen to be on balance the most highly expressed, the most conserved, and they've been chosen to span the highest number of clinically important variants. And only very recently, the main collaboration released additional set of transcripts called the main plus clinical. There's a rare, there's only very few of them. There's 43 of them genome-wide so far. And maybe these are not the most representative, but they're important as there might be some clinically important variants, so pathogenic variants, not captured for the main select transcript. And if that's the case, then additional main plus clinical transcript is introduced to capture this missing clinical variation. So the purpose is to enable the clinical reporting of such variants if they're not covered or captured by the main select transcript. And here we have an example. And don't worry if you cannot read it, it's not relevant. I just wanted to point out that here I'm showing you first the Ensembl gene at the top of the page. So those transcripts highlighted in green indicate the Ensembl gene, and you can see there's a couple of different alternative transcripts. We then have a ref-seq gene, so you can see there's also many alternative transcripts, but there's only one main select transcript for this locus, which is the most relevant and the most representative. And in this rare case, we also have an additional main plus clinical transcript. So why do we have the main plus clinical? Let me just zoom in to explain that further. So here we have our main select transcript in blue, and the main plus clinical is shown in red. And you can see they have mutually exclusive exons. And there is some clinically important variation over here indicated by those bars, which is not captured, which is not covered by the main select transcript, but it falls in the main plus clinical transcript. So I hope that's clear. And people often ask us, which transcript is the one? So which transcript do I choose to work with if I'm doing some experimental work, if I wanted to design a CRISPR knockout? And there's no straightforward answer, as all of those transcripts are real and valid, and they might serve different biological functions, they might be expressed differently, but we can provide a guideline on how to choose the most biologically relevant or representative. So if it was up to me to choose one transcript to work with, I would follow this hierarchy. So I would first look out for a main select transcript, which we just covered. But then if main select annotation was unavailable, then I would move to APRIS. So APRIS is another transcript scoring algorithm developed by a group in Spain. And those guys, they use their own judgment. They look at slightly different they have slightly different priorities, but it's a similar focus. So they look at protein structural information and presence of active sites, but also cross-species alignment, and score transcripts from 1 to 5, with 1 being the best. So 1 stands for P1, the principal isoform, but there are some additional ones as well. So if APRIS wasn't available, I would then move to GenCode Basic, which is basically 5' and 3' complete GenCode transcripts. So what does that mean? So now we know that we annotate our transcripts based on real experimental biological data, and biological data might be sometimes incomplete. So we would still make a transcript annotation, even when it seems that the transcript might be incomplete, so it might be missing either 5' or 3' bit. And those incomplete transcripts are then filtered out for a GenCode Basic. We then have the transcript support level, which also scores transcripts from 1 to 5, with 1 being best. We then have CCDS, which stands for Consensus Coding DNA Sequence, which is a little bit like main, but a little bit older. So it's also an agreement between NCBI and Ensembl, but only for coding sequence. So it doesn't take the UTRs into account. And finally, we also have the golden transcripts, which we just covered as well. We talked about them, but by no means these are the most important ones, but they're useful if there's nothing else out there. And someone asked, how do you determine clinically important relevant variants? So we don't do that. That comes from the scientific and clinical community. So if there is evidence that the variants are clinically relevant and been shown to be causal of certain disease and phenotypes, pathogenic phenotypes, and then we refer to them as clinically important. Someone asked, can you elaborate a bit on the transcript? If you could ask me a more specific question, that would be great. I can get back to it later. All right, so let's move on with the lecture. So another linked topic is the Ensembl canonical transcripts. So Ensembl canonical transcript is a single representative transcript identified at every single locus. Some analysis might require selecting only one transcript per locus, like for example, calculating of Ensembl gene trees. So for human protein coding genes, the canonical transcripts are going to be transcripts which on balance are the most highly conserved, the most highly expressed. They're concordant with Apris P1 isoform and a UniProt canonical isoform. They have the longest coding sequence and cover the largest number of clinically important variants, and they should be complete. And for human protein coding genes, Ensembl canonical is pretty much the main select transcript, so the most representative. And you can find all of these transcript flags and scoring algorithms in a transcript table in Ensembl. So I'm here showing you transcript table for BRCA2 gene. So transcript table lists all transcripts of a given gene. And here you can find this information that we just talked about in this columns shaded in green over here. So you can find the biotype, which is color coded. You can find the corresponding CCDS annotation, UniProt match and RefSeq match for a main select transcripts. And we have feather flags like the main select, canonical, GenCode basic, Apris, TSL, and so on and so forth. All right, so two more concepts before we move to the demonstration. All of our features have Ensembl stable IDs assigned to them. So Ensembl stable IDs are composed of these ENS prefix, and then comes a letter indicating the feature type. So we've got G for genes, T for transcript, P for proteins, E for exons, and R for regulatory regions. It's not an exhaustive list. We have other types of stable IDs like gene tree stable IDs or protein binding motif IDs and so on and so forth, but these are the most important ones. And then comes the 11-digit unique number and a versioning after the decimal point. So why are these important? I know this might not be the most meaningful. There's a bunch of, it's a long number, which might not mean much to you. You are more likely to be working using the gene names. However, bear in mind the gene names are not very unique and are not very stable. So gene names do change. There are many synonymous gene names, while the Ensembl stable IDs shouldn't change between releases. If there's a small change to the underlying feature, we might then increment the versioning, and we have a clear definition of what a small change is. But if there's a larger change to the underlying feature, we would then retire given stable ID, and we would issue a new one indicating that it's a different feature from now on. And for non-human species, there's also a suffix added to indicate the species. So for example, for a mouse, you're going to find moose. For a zebrafish, we have dar. And if you wanted to learn more about Ensembl gene annotation, please have a look at our publication. And before we move to our demonstration, let's talk a little bit about gene ontology terms. So human language is ambiguous. We have many synonyms and multiple terms for the same thing. So we might end up using slightly different language to describe the same thing. And it's the same in the scientific community. So for example, when talking about innate immune response, one person might be calling it innate immunity, while another person could be calling it non-specific immunity. And we know they mean exactly the same thing. However, the computer searchers do not cope very well with that. And to get around that problem, the scientific community came up with a gene ontology term, so which is basically the standardized, controlled, and easily searchable vocabulary to talk about gene function and to make sure we're talking about the same thing. And here we have an example. So they all have the accession numbers, name, type, and a definition. And they organize in hierarchy, which means a given term, like for example, innate immune response might have a more general parent term, like for example, immune response, but also a more... Sorry, I just muted someone who just joined. And more specific child terms, and there could be quite a few of those, like in this example. All right, so we're now going to have time for our demonstration. Let me just go back to the questions. I think there were a couple of questions asked. So someone asked to elaborate on the main transcripts. As I said, Ensembl and NCBI have their independent gene annotation pipelines. And then we collaborate closely with NCBI to come up with this list of more of the most representative and relevant transcripts per locus. So we're trying to find transcripts which are identical between NCBI and Ensembl. And we also use all sorts of experimental data trying to predict which transcripts could be the most relevant when it comes to their expression level, conservation, and also reporting of clinically important variants. And so, Mas, is there a reason for different number of isoforms in NCBI Ensembl? Yet again, we have slightly different pipelines which work slightly differently. So we take into account similar priorities. But as there is a difference in computation, then you'll find slight differences. Furthermore, we annotate transcripts on the reference genome, so our transcript have to match the reference genome, while NCBI allows for mismatches and annotates transcript directly on mRNA. All right, so if there are any other questions, please feel free to add them to the living document. I'm gonna reply to those questions during the exercises. And now let's move to our demonstration. So we're going to look at an Ensembl gene and try to find information about this gene and its transcript. So let me just jump out of my presentation now. I'm gonna copy the gene name. And I'm going to go to my internet browser. So I'm here on the Ensembl homepage, and I'm going to paste my gene name into the main search box and click Go. So as I didn't specify which species I'm interested in, I didn't choose a species from the species selector. I got search results across all taxa, with the most popular hit at the top of the page. So we have a human gene on top. I could further restrict my search results using the categories on the left. So I could restrict them to a given topic or species of interest. As I'm interested in the human gene, I'm going to click on the gene name to go to the gene tab now. And that takes me to the gene tab, which is the second type of tab we're exploring. We explored the location tab last week. So here we have the gene tab opened, and you can see the location tab is opened just next to it by default. So that's where the gene sits in the genome. And the gene tab is dedicated to genes. So it collates information about genes. You can find the tab specific menu on the left. So this menu lists all available views, displays and in this tab. So we've got this gene-based displays. We then have some tool buttons below. And by default, we land on the gene summary page. So we first have a little overview. So some overview information about this gene. So we've got the gene name and the Ensembl stable ID. We've got the gene description, some synonymous names, but also the gene location and the strand information. And below you can find the transcript table, which you can hide and show. So this table lists all transcripts of this gene. You can find there the transcript name, which is arbitrary. I wouldn't worry too much about it. We then have the transcript stable ID, which is way more important. We have this transcript length, and also the protein length of the translation for a coding transcript. And then we have all of the columns and transcript flags we talked about in our lecture. And all of those, if you can see this dotted underline, that means you can hover over and there's a bit of text to get a bit of more information and explanation. So if you don't remember what any of those mean, just hover over or mouse over it to learn a little bit more. And this little overview is available on every single page in the gene tab. So if you go to subsequent gene pages, you'll always find this little overview at the top of the page. And below we have a page specific information. So we have more summary information, which also includes the annotation method, and we have a graphical representation of this gene and its transcripts. So I now wanted to show you a sequence view. So I'm going to scroll up and go to sequence in the left-hand side menu. So this view shows this sequence of my gene of interest in the flanking region. So you can see the sequence here is given in FASTA format. We have the FASTA header line, which starts with greater than symbol. We then have some information about the genomic coordinates. So we've got the genome assembly, we've got a chromosome information, start position, end position, and the strand information. So 1 stands for forward, -1 would stand for a reverse. And then you can see in the legend that all exons are highlighted in this peach color. And the exons of my gene of interest are further indicated by this brownish lettering, okay? So what are those exons with black lettering? Well, those are basically exons, overlapping exons of neighboring genes. And then you can blast the entire sequence from this view by clicking on this button. Alternatively, you can also blast the selection. So if I highlight a sequence here, I'm going to get this pop-up option to a blast selection. And you can blast a sequence pretty much from any sequence view in Ensembl. You can customize and manipulate that view by going to configure this page in the left-hand side menu. So let's click there for now. And here I can, for example, modify the flanking sequence. So let's say I wanted to see shorter flanking sequence. So I'm going to change that to 50. I could add variants to this view. So I'm going to choose show variants yes, and show them with links. We could also filter, excuse me, filter variants by consequence. And so I could, for example, choose to only display missense variants, but I'm not going to do that for now. And finally, I can also add line numbering. And once you're ready choosing your options, simply click on the tick, save, and close. And that's going to update the image. So the markup is loading. It might take a little bit, depending on how much variation data there is, but there's usually quite a lot of variation data. So you can see the flanking sequence is now shorter, and now we have a bunch of genetic variation visualized in that view on the sequence, and the variants are color coded. So the variant consequence legend is found at the top. So for example, the missense variants are shown in yellow, the intronic variants are shown in blue, and all of those features here are interactive and clickable. So I can click on a given variant to get a pop-up menu, a little bit more information about this variant. And then Elena will introduce genetic variation in more details during our next session. Finally, you can also download this sequence by clicking on this blue button at the top of the page. So let's click on a download sequence now. And here we have a pop-up window. You can choose to download either FASTA sequence or RTF sequence, which stands for a rich text file, which is pretty much just a static image with all of its decoration, annotation, and coloring, which can be opened with the word a processor, so you can use it for your figures or lab book and notes. You can then decide what type of sequence you'd like to export. So by default, we have a genomic sequence being exported, but you can change and you can choose from multiple options. You can also further modify the flanking sequence here. You can then preview your sequence, and you can choose to download compressed version as well if you wanted to. So let's close this window for now by clicking either on the tick or anywhere outside the box. So let's now talk a little bit more about gene function. So if you take a look at the menu on your left, you'll find that the ontology is split into 3 categories. So we've got biological processes, which tell you what the gene does, molecular function, which tells you how it does it, and cellular component, which tells you where the protein is located in the cell. So let's click on biological processes for now. So this table lists all biological processes associated with this gene. So in this table, you can find the gene ontology accession, number, the term name, and the evidence. You can see you can hover over to get a bit more information about those evidences. You can find the annotation source, but also transcript ID. So why do we have transcript IDs here, but not the gene ID? Well, it's because the gene ontology terms are assigned on a per transcript basis. So they're associated with transcripts or translations. And this particular table isn't that large, but they can get quite massive, believe me. So you can further filter this table, and you can download it using this Excel sheet icon. And another view which on this tab, which I wanted to show you, is a gene expression view over here. So let's click on gene expression now. So here we display external data from the expression atlas. And it has this really nice interactive widget. So you can highlight different tissues and organs. You can change between sexes, but also different brain regions. And if you highlight certain tissue, it's also going to highlight the corresponding column in the heat map next to it. So here we have the heat map, which summarizes this gene's expression across different projects and different tissues. and the expression level is indicated by a color. So the darker the color, the higher the gene expression is. And you can further filter this data and download it. And the last thing which I wanted to show you in the transcript tab is the external references view. So let's now click on the external references. And this page links you out to external databases, which also have some records and information about this gene. So you can see we'll link out to Expression Atlas, but also OMIM, NCBI. You also have a table with some corresponding IDs in external databases. So it's a really great place to branch out as well. All right, so that's about it when it comes to the gene tab. Let's now explore the transcript tab. But before I move to the transcript tab, let me have a look at the questions. You can see there's a couple of questions in the chat box. Is there information about epigenetic state of regulatory regions available to, for example, methylation, acetylation marks, and histone state promoter? Yes, we do have regulation data. We have separate tab dedicated to a regulatory element. So if you can maybe show you quickly, if you go to the location tab, you'll find the regulatory build displayed in a separate track. So here we have, for example, promoter indicated in red. So if I click on this promoter stable ID, that's going to take me to the regulation tab. We're not covering that in detail throughout this course. That's why I'm mentioning very briefly now. But here you can find lots of information about epigenetic state. So if you go to details by cell type, you can add different tissues to this view and experimental data, including histone modifications, transcription factor binding sites, and DNA's hypersensitivity. We also have some DNA methylation in the location tab available as a track. What is 1 in FASTA header line? So a 1 or -1 represented the gene strand. 1 stands for forward strand, and -1 for reverse, as someone just explained. And someone asked why transcript ID instead of a gene ID. It's because I believe you're referring to the gene ontology terms. It's because gene ontologies are assigned to transcripts. So for example, you can see those two transcripts here are very different. So for example, one of them codes for a long protein, while the other one is not very functional and is non-coding. So they're going to have a very different function. That's why gene ontology terms are assigned on a per transcript or a per translation basis. All right, so let's now move to the transcript tab. So I scrolled up to the transcript table, and I'm going to go to the transcript tab by clicking on the transcript ID hyperlink. I'm going to choose the main select transcript, which is the most relevant or representative, if you wish. So if I click here, that should open the transcript tab. So you can see now we have the transcript tab open next to the gene tab. So you can toggle between them. And again, by default, we land on the transcript summary page. You can find some summary statistics about this transcript, including the number of exons and the transcript length, but also annotation method. Here we have the transcript graphical representation. And the first view on this transcript tab that I wanted to show you is the exons view under sequence in the left-hand side menu. So let's click there. And this view lists all of the exons of this transcript summarized in table. So you can find the exon rank in the transcript, but also the exon stable ID, the genomic coordinates and the exon phase, but also length for both exons and introns. The sequence here is color-coded. You can find the explanation in the legend above. So the flanking sequence is shown in green. We then have UTR sequence in orange and a coding sequence in blue. So you can see the coding sequence starts with ATG and finishes with TGA, which is a stop codon. And then intronic sequence is shown in gray and it's by default truncated. So yes, again, if you wanted to customize and manipulate this view, you can go to configure this page. And here we can change the length of the flanking sequence. Imagine I wanted it longer. You can also show variation in this view, but I'm not going to do that for now. And you can also show a full introns as by default they're truncated. So I'm going to expand them by ticking this box and then save and close. And now you can see we have a longer flanking sequence and my introns are shown in full. Great stuff. So let's scroll up and explore the cDNA view now. So cDNA is a spliced mature transcript. So transcript without introns. Let me just switch off the variation data. I think it's going to be much clearer if I'm going to switch that off. So this sequence view is composed of 3 lines. So the first line is the actual cDNA, which includes UTRs shown in this dark yellow color. The second line is the coding sequence. So it starts with ATG, which is a start codon, and ends with TGA, so the stop codon. And then the third row shows you the corresponding protein sequence. So it starts with methionine and finishes with stop. Then different exons of our transcripts are indicated by this alternating black and blue lettering. So you can see that the first exon finishes somewhere here, and then we have the second black exon, and finally the codons are indicated by this alternating yellow highlighting or the lack of it. Great stuff. And before we move to the protein information, I'll quickly wanted to point out, bring your attention to the external references section in the menu on the left. So here it's split in 2 categories: general identifiers, which is very similar to the external references that we explored, that we just saw for the gene tab. And we have additional view named oligoprobes. So let's go to the oligoprobes now. This view lists all microarray probes which map to this transcript and can be used to measure the expression level of this transcript. All right, so before we move to the exercises, I just wanted to talk a little bit about protein information, which you can find in Ensembl. So proteins are not features in Ensembl, as they do not map to the genome reference directly. They map to the genome via transcripts. We don't have special dedicated tab to proteins, but you can find some protein information in the transcript tab. So let's first explore protein summary view. So I'm going to click on protein summary in the left-hand side menu. And here you can find the graphical representation of this translation. So here we have the N terminus end, and here we have the C terminus end. This protein is colored by the exons. So we have our 2 exons, first shown in this pink color, second in purple. You can find some protein domains and motifs predicted by different in silico methods. So these are all computational predictions. They haven't been functionally validated, but if a given domain or motif are predicted independently by different methods, are more likely to be real. If you're not a fan of graphical diagrams, you can find exactly the same information summarized under domains and features. So let's navigate to this view now. And here we have a table listing and different domains falling in this translation. So you can find the domain, source, position, and description, accession numbers, and some links to external resources. And the last view, which I wanted to show you, is the 3D protein model. So we import this data from a PDBE, and it has got this really nice interactive widget. So here you can see our translation with some additional proteins. You can rotate it, you can zoom in and zoom out. You can also change a protein model. So here, this drop-down list gives you top PDBE hits for this particular transcript. And then there's also some additional Ensembl functionality. Let me just zoom out a bit to show you this. I think it's going to be a little bit easier when you can look at the image. So first of all, I can visualize my exons of my transcripts on this model. So now you can see our two exons are shown in different colors. So we have this green and this purple exon. You can see that this translation is actually part of a larger protein complex, and the other proteins are shown in gray. You can also visualize some protein domains in that view, and also pathogenicity predictions for a variant falling in this translation. Okay, great stuff. I mean, there is more functionality, there's more to explore, and now we have time for your own exploration. There are some exercises we might ask you to explore certain features which we haven't covered in the demonstration, but give it a go, explore, click around, and then if you have any questions, please let me know. So you can find your exercises on page 32 in your course books. I'm going to be here answering the questions in the living document now. And also, if you struggle with any of the exercises, please let me know. I'll be very happy to go over the answer on the screen and explain how we got there. Crack on. Sure, we have a question about exercise GT3. Let me have a look. I was going to jump out of this. Let me open your course books. So I'm going to go to exercises, genes, and transcripts, and that was exercise G3. So we're exploring a gene in plants, and we are given gene ID. So here we have a gene name, here we have a gene ID. I'm going to copy this gene ID. And I'm going to go to Ensembl genomes. So as we were working with the plant species, I first navigated to Ensembl genomes, and from here I'm going to navigate to Ensembl plants. And I'm going to search for my gene of interest by typing the gene ID in the search box and clicking go. There's only one hit returned, so I'm going to go to the gene tab by clicking on the gene ID hyperlink. And if I click on show transcript table, that's going to expand this table and show me all of transcripts of this gene. There's only one. So let's go to the transcript tab by clicking on the stable ID. And someone says that the summary says that this transcript has 8 exons, indeed. And there was a problem with the graphical summary. So let's have a look. I can see 1, 2, 3, 4, 5, 6, 7, 8. So it seems to be matching. We've got 8 boxes, and that matches the reported 8 exons. Sometimes, depending on your browser, if the window is too small, those boxes kind of merge together and it's difficult to tell them apart. That's why it's always better to navigate to this exons view, because then you're going to see the exons summarized in table. So you're not going to miss anything. And you can see here we also have 8 corresponding exons. So I hope that helps. If there are any other questions regarding your exercises, please do let me know. We still have about 15 minutes left. So there's another question about the G2 exercise. So let's have a look. As we're still on this page, I just wanted to add an extra comment because I think the confusion arose from the fact that those last exons, so pretty much, you know, the 3' farmost and 5' farmost exons are empty, which means they're UTRs. That's right, they're UTRs. They're non-coding, they're not translated, but they're still exonic. Okay, remember, UTRs are also exonic as they remain in the transcript after splicing. So they're exons too, so we have to count them. So let's move to the G2 2 exercise. So I think you're asking about the OMIM gene identifier for this gene, right? So we are asked to search for a given phenotype and then find associated gene, and then we are asked for corresponding OMIM gene ID for this particular gene. So let me just copy the phenotype name. I'm going to go to the Ensembl homepage by clicking on the Ensembl logo. And then I'm going to paste my phenotype name, and I'm going to click go. Now I can click on the phenotype name to go to the phenotype tab, which should list all loci associated with a given phenotype. You can see there are different types of loci, including both variants, structural variants, but also genes. I can turn all off, and then I'm going to choose genes only. So there's only one gene associated with this particular phenotype. Let's click on the gene name to go to the gene tab. And remember when I showed you the external references, this is the view that lists corresponding entrants on that gene in other databases. So if I go to external references and scroll down to this table over here, you'll find the OMIM gene information, including the corresponding OMIM gene ID. So that's this number we are asking you to find. So I hope that makes sense. Are there any final questions? As I can see we only have 5 minutes left. Also, please feel free to keep adding questions to the living document. Even once we finish, you're more than welcome to keep adding them, and then we'll address all the questions during the next session or even between the sessions. I'm going to wrap up for today in about 3 minutes, so there is still a chance for last minute questions. All right, everyone. We only have 2 minutes left, so let me just tell you what to expect from your next session. So this Friday, I believe, 25th, you will have variation and develop with Elena. So Elena will introduce the genetic variation data you can find and explore in Ensembl. And you'll be also talking about the Ensemble's variant effect predictor, which is a really nice and handy tool to annotate your own variation data. That's all from me. Thank you very much for joining. Also wanted to thank our hosts for having us today. And please feel free to keep asking questions and we'll address them in a due time. Thank you very much for joining, everyone.
So thanks, Michelle, for your sessions. And with these words, now I seek your permission to end this session.
Thank you very much.